All right, it looks like our number of attendees is pretty stable. So I guess I'll start out. So welcome everyone to our Armor seminar for this week. It's my pleasure to introduce Dr. Spencer Hellert, who is a postdoc here at the museum, supported by an NSF grant, and also currently is doing some teaching at Columbia College, Chicago. So she's actually coming to us live from her office on campus there. In terms of her background, Spencer did her undergrad at Augustana College in geology and biology. And she actually worked with Bill Hammer there, who was in the geology department and has done a lot of field work in Antarctica and has been a longtime collaborator with Pete Makovicki in the sort of paleontological research that's been going on at the museum in Antarctica. It's kind of an interesting connection there. After that, she did a master's degree in ecology, evolution, and conservation biology at the University of Illinois in Urbana-Champaign, and then went on to do her PhD in geology and had some interest in biology as well at Indiana University, where she worked with David Pauly, who is a well-known paleontologist and morphometrician. So Spencer's work, as she likes to often put it, is turning shape into math. So she does a lot of morphometrics work and has been very interested in questions about using morphometric approaches to look at morphological integration and its effects on evolvability. And I think that's some of what she's going to be talking about today. And the work that she's been doing with me in the context of our NSF grant has been more focused on synapsid evolution and using morphometric approaches to look at the evolution of shape and function in synapsid forelimbs. And also part of that's kind of taken way longer than I think either of us has anticipated. She's also been in the process of making like the world's largest synapsid phylogeny. And so that's almost done. We're almost ready to submit a manuscript on that. And so yeah, Spencer, I will let you take it away.
Thank you so much. I'll go ahead and start sharing. And whoops. Like that. Let me switch this display around. There we go. I think that looks good.
Spencer, you actually, sorry to interrupt, you actually had the other way around. Right now we have like the presenter view.
Just kidding. There we go. How's that? Okay, so title of my talk today is because of the way that it is, connecting genetics, development, and morphology by quantifying shape. So just very briefly to understand how all of this works. So we're all taught when we first are taught about evolution that DNA is the instruction book for how we build an organism, and that changes to the genome lead to differences in organisms. We also learn that those differences can lead to individuals with traits that undergo selection because they allow the organisms to survive better and have more babies that also survive better, which results in a change in allele frequency in the population and thus evolution. We also learn that natural selection in concert with extinction has produced a spectacular array of forms that are adapted to a whole range of environments. This amazing insight has been rightly celebrated for over a century, but of course, evolution is a little more complicated than that because the simple formula of variation plus selection plus time equals evolution of form doesn't explain why there hasn't been instances of truly intense, kind of insane levels of evolutionary change. So after all, with this formula, given enough time, one type of organism technically should be able to basically evolve into any other organism. The classic hypothetical example here being a horse turning into a fly. Maybe it's because there just hasn't been enough time to see those kinds of evolutionary changes, but we know that probably isn't true because some populations have actually undergone a great deal of evolution pretty quickly, the classic example being domesticated foxes. So a quick thing before I change slides here is that I remember asking these types of questions as an undergrad and getting the response that basically it's just the way that it is. And so my research program aims to understand why organisms go down some evolutionary paths quite quickly while other evolutionary paths seem inaccessible, so that I never have to explain evolution like this.
Oh, wow, score! Look at this. This is an aspen. You can tell that it's an aspen tree because of the way it is. Wow.
So specifically, my research is on how morphological traits can be integrated in ways that can sometimes facilitate and sometimes prevent evolution from occurring in certain trajectories. Trait integration is when one trait cannot evolve without coordinated change in another trait. And integration is caused by genetic, developmental, and functional factors. One example of a genetic factor is pleiotropy, which is when the same gene controls multiple traits. This is a totally fake example, but I love using it because for visual purposes, it's easier to understand. So let's say that the same gene controls whether or not you have a head and also the size of your feet. And for whatever reason, it would be really advantageous for humans to have big feet. But since the same gene controls foot size and the presence or absence of the head, if humans have big feet, let's say they don't get a head. You can't exist without a head, and so you will never be able to evolve large feet. And so again, this is a fake example, but a way in which sort of a road down an evolutionary path is completely blocked. In terms of developmental processes, they're often very closely related to genetics because genes set development in motion. But here are two examples that don't necessarily involve genes. So this video shows very early development. As all of you probably know, we start out as a big ball of cells, which becomes more complex over time. One chunk of this ball may become several different structures in the body. So whatever happens to that particular chunk of cells early on will influence multiple traits. Below the video is an image of a chick inside of an egg just before hatching. And it's really, really scrunched up in there. If the bones of the wings are too long or not in the right proportion, the chick isn't going to be able to fit inside of the egg correctly, which might hinder the development of other parts of the body or prevent the development from finishing in the egg completely. Lastly, multiple traits or structures can be involved in the same biomechanical apparatus. So imagine that the lower beak of this bird evolves to be very long. Then it won't be able to eat very well, and it won't survive. And the upper and lower beak, therefore, cannot be evolutionarily independent. So a tension therefore exists between the processes that promote anatomical diversification and the processes that promote trait integration. My work helps connect the dots between genetics development and adult morphology in order to understand how trait integration affects morphological evolution. And I do this by quantifying shape. I've done this in several different ways, and I'm going to describe some of those projects here. So first, I'll talk about some of my PhD work on flightless birds and a little bit about horned dung beetles. Then I'll talk about my postdoc work on synapsid forelimbs, and finally, I'll describe some near future work on synapsid jaw and middle ear evolution. So for the first project I'm going to talk about, I assess the extent to which integration has constrained the diversification of the limb elements in birds across the many times that they have evolutionarily lost the ability to fly. The transition from flight to terrestrial locomotion occurred time and time again as birds have become secondarily flightless across the avian clade. This offers natural repetition of this transition to a terrestrial lifestyle. And flight is really expensive, and because for the most part, birds retain the locomotor function of their hind limbs after gaining the ability to fly, the forelimbs are free to possess a locomotor function or not. Thus, flightlessness is pretty common. For modern species, flightlessness has occurred in at least 26 families and in 17 orders. Forelimbs are very useful things. Since flightless birds no longer have functional pressure on their forelimbs to fly, they should basically be able to do whatever they want with their wings, and yet they don't.
Oh my god, oh my god, he's gonna cut, he's gonna shoot, he's gonna shoot me,
This is especially strange because sexual selection is a really big deal in birds, and male birds across the bird family tree use everything they possibly can to tell females that they should choose them. Birds can't do much with their wings in terms of sexual display because they have to stay usable for flight. But you would think that once they don't have to fly anymore, birds would utilize these conveniently mobile, large, and functionally liberated appendages for sexual display. Ostriches use their wings a little for sexual display, but flightless birds, don't use their wings for almost anything. We wanted to know if the similarity of flightless bird forelimbs was possibly due to a genetic or developmental constraint in the forelimbs of all birds. So I looked at the hind limbs as well, but I'll focus on the forelimbs here. We expected that the biomechanical demands of flight would lead to an evolutionary constraint in flying avian forelimbs, and that in the absence of these demands, forelimbs would be free to evolve in different directions. When we look at our results, a functional driver of covariance would result in different covariance patterns in the limbs of flying and flightless birds, while a developmental or genetic driver of covariance would result from similar covariance patterns in the limbs of flying and flightless birds. Integration can be broken up over evolutionary time into pockets of tight integration so that each group of tightly integrated traits have to evolve together, but each group can evolve semi-independently of each other. So in the image here, each color represents a different pocket of integrated traits in the skull. And these pockets of integration are called modules. How tightly traits are integrated and how modular the pattern of integration is can be measured by quantifying patterns of variation and covariation in the traits that make up an anatomical form of an organism. Okay, so Ken told you that I like to say that I turn shape into math, and it's more simple than you think. And to get at the factors underlying integration within these bird forelimbs, I had to do that. I had to turn shape into math to quantify integration patterns in the limb bones. And like I said, it's not actually all that difficult. So if you think of sort of how facial recognition works, it's a little bit like that, where you take whatever object you're interested in. So in this case, it would be a face for this example. And you find points on that face that you can find on every other face you're going to look at. And you place landmarks at those points. And really, that's a 2D picture made of a bunch of pixels in a grid. And so each landmark lands on a pixel that has a particular X and Y coordinate within that grid of pixels. And so your list of coordinates for your landmarks is turning the shape into math. So the first thing I did for the bird limbs was take digital photographs of the three major skeletal limb elements in the forelimb, the humerus, the ulna, and the manus. There. Then I took 3 to 6 landmarks and an outline of semi-landmarks for each bone using TPS Dig. Then the outlines were broken up into segments of semi-landmarks using the original homologous landmarks. So in total, I had about 200 semi-landmarks for each of the limb bones. The landmark configurations for each type of bone were then superimposed using Procrustes superimposition. So for Procrustes superimposition, the landmarks of the same bone from every specimen are translated so that they lay on top of one another. Then they're scaled to the same size and rotated so that all the landmark configurations are in the same orientation. The new Procrustes landmarks are then used to create variance covariance matrices, which then can be used in various analyses to see how landmarks wiggle around in space from species to species and how landmarks wiggle around in correlation with other landmarks. I conducted a whole series of different analyses to look at within bone integration and between bone integration. Each of these tests only gets at certain aspects of evolutionary patterns of integration. So we did an array of analyses to get a holistic picture of the evolutionary patterns of integration in the forelimb. This is one of the cluster analyses for the manus of flying birds, which allows us to visualize the modular organization of integration within bones. So each branch on this dendrogram produced by the cluster analysis represents a landmark on the bone pictured on the right. The analysis clusters the landmarks based on how tightly they covary. So in this case, the landmarks are split between 2 modules of integration. And then I color-coded the landmarks by the module that they belong to. The overall pattern of integration within each of the limb bones was very similar across flying and flightless birds. I'm showing the mantis here, but it's true of all three of the limb elements. These are PCA plots for each bone. The plots show the two principal components of shape change that account for the most amount of change across all bird species. So flightless birds are in blue and then flying birds are in orange. There's enough overlap in each of the plots to say that the patterns of integration within each of the limb bones is significantly similar in flying and flightless birds. These are the 2-block partially squares plots that we did for each forelimb, and we also did them for the hind limb bone pairs as well. A 2-block partially squares is a lot like a PCA, but the axes represent the axis of greatest variance for each of the elements involved for each plot. The lines are then just best fit lines through the flying and flightless data points to help visualize the data. So in these plots, you can see that the flying and flightless taxa are generally somewhat separated in the morphospace, but the pairs that make up the two most distal elements in, so the middle plot in this case, are especially mixed together. When we look at the array of tests for integration pattern similarity within each bone, we found that all of the tests point to flying and flightless birds having similar integration patterns. Allometry didn't affect the results, and phylogenetic relatedness didn't either. When we look at the array of tests of integration pattern similarity between each pair of bones, we found more mixed results, but in general, it looks as though there does seem to be a difference in patterns of integration between bones, and that phylogenetic relatedness can make it seem like there's more integration than there really is, so we took that into account. Instead of just looking at integration within each bone and between pairs of bones, we also wanted to look at what's going on in the limb as a whole. And so to do that, we placed each of the elements from the same limb in the same coordinate space and did a cluster analysis again. So by doing this, we're able to look at the covariance within, between, and among all of the bones at once. The results reflected what we had found from the other analyses. So here you can see that the patterns within each bone are slightly different, but the overall pattern of covariance within each bone is very similar in both flying and flightless birds. And if we look at the areas of covariance that are shared across all three bones, we see that different areas are involved in both flying and flightless birds. The covariance patterns within, between, and among all of the bones all contribute to the overall pattern of integration in the limb as a whole. Studies that analyze covariation using only individual elements or only pairs of elements may miss the overall pattern, and therefore misinterpret the development or genetic and functional factors that underlie integration. So placing multiple major elements of the limb in the same coordinate space allows the evolutionary pattern of integration of the whole limb to be better understood. The results partially support our hypothesis that the apparent homoplasia of flightless bird limbs is due to a genetic or developmental constraint acting on all birds, despite very different functional requirements of their limbs. In the absence of the functional pressure of flight, the forelimbs are reduced because of the immediate benefits of no longer maintaining such an expensive apparatus. However, the results suggest that in addition to this, a developmental or genetic constraint on within bone covariance may restrict the evolutionary potential of forelimb morphology as a whole. We also looked at being able to tell apart birds based on flight style in morphospace, and it turns out using the same set of landmarks that you can tell the difference between different major flight styles, and that flightless birds do take up more of the shape space, but when combined with our previous results that the integration patterns within each wing element are significantly similar in flying and flightless birds, it supports our conclusion that all birds' wings are under a genetic and or developmental constraint as a whole. Okay, so moving on from flightless birds, I'm going to jump over to horned beetles for just a moment. I promise it connects. So I worked on many different kinds of taxa for my PhD, including birds, not avian theropod dinosaurs, and these dung beetles. So like I said, I'm not going to dive into these for very long, but I want to mention it because it shows kind of the progression between doing landmarks in 2D versus landmarks in 3D. So some of these beetles have horns on their heads, which are evolutionarily novel structures. The adult beetle rolls up a dung ball and shoves it down a tunnel, and then the eggs are laid in and around the dung ball. And then when the babies hatch, they eat the poop. And usually the males are the ones that have the horns, and whether or not the males have horns is totally dependent on how much poop they eat as a baby. So if they eat enough poop, they get big horns. If they don't eat enough poop, and it's a really strict threshold, then you don't get any horns. There's my little video. There we go. So it turns out that within this Onthophagus genus of these beetles, there's a lot of different things going on with horns on the head. Some of them don't have horns at all in either sex. Some of them, the females don't have horns, some of the males do, but there are both males with horns and then sneaker males without horns. And then lastly, there's this really interesting species called Onthophagus sagittarius, where the female has horns, but it has one horn in the middle of the face, like a little unicorn horn, and the males have small horns at the front of the face. So the horns are changing shape and they're changing location all over the heads of these guys throughout the genus. And so using that, you can ask some really interesting evolutionary questions, especially because I was lucky enough to work with Armin Moschek's lab at IU, and he was working on the development of these head horns. So he was figuring out what genes are responsible for the horns, and it turns out that when you mess up the genes, you don't get horns, which is expected, but some of these beetles got eyes where their horns should have been, and the eyes work. So using that information, you can start to connect the dots between what's going on with the genes, what's going on in development, and what's going on with the adult morphology. And then you can sort of place into that trait integration and also region identity within that whole scheme. Okay, so now I'm going to talk about my current postdoc work, which is the synapsid forelimb evolution grant. So as Ken said, I was hired through an NSF grant that he got with colleague Stephanie Pierce at Harvard, and it's to investigate synapsid forelimb evolution. Synapsids are a group that consists of mammals and their ancestors. So mammals can run, climb, jump, hop, swim, swing, dig, etc. So as a group, they can do more with their forelimbs than any other group of animals, and we would like to figure out why. Part of this investigation involves computer models of joints and muscles to figure out how it's possible for the total range of movement within each kind of synapsid through the clade. And my job within this project is in two parts. First, in order to figure out how forelimbs change across the synapsid clade, we need a phylogeny. So my first job, as Ken mentioned, was to build a comprehensive synapsid phylogeny. So instead of a traditional phylogeny, I created a metatree, which I'll explain in a moment. And then using that phylogeny, I will quantify the shape of the forelimb structure across the synapsid clade so that I can then analyze how forelimb elements change through evolutionary time. Okay, so this metatree, it's explained in detail in a couple of papers, including Lloyd et al. 2016 and Lloyd and Slater 2021. So I'm not going to go into a huge amount of detail here. But essentially, a metatree is a phylogeny that shows the consensus of evolutionary relationships through the literature. So the first thing we did was collect every single morphological character matrix from the literature as of July 2021, where non-mammalian synapsids compose the majority of the operational taxonomic units, or OTUs. We also collected the most comprehensive early mammaliaform matrices and added those to our data set. So each of the 211 matrices we ended up with was weighted according to its publication year, and it's dependent on parent matrices. So often new phylogenies are constructed using older character matrices with the addition or alteration of a few columns of traits. So a lot of phylogenies are just slightly different versions of previously published phylogenies. So if you map parent-child relationships among all of these matrices and then weigh them accordingly, you can avoid certain base matrices overpowering the data set. Our metatree is at the species level, and unfortunately, many of the OTUs in the matrices were at the genus level or above. So something I had to do was find out what exact species the authors used to describe the traits in their matrix, and that was not an easy task. Equally time consuming was going through the paleobiology database to fix taxonomic issues and add any missing synapsid taxa. So far, myself and my colleague Dave Grossnickel have made, we think, over 800 changes to the PBDB. Using these 211 published matrices, we produced a metatree of 2,875 synapsid species. To time calibrate the topology, we queried the PBDB to obtain occurrence data, and where possible, we used biostratigraphic radiometric age estimates from primary sources. So reliable occurrence data were obtained for roughly, I guess, over 1,800 of the taxa in the phylogeny. We used the time paleo5 function within the paleotree R package and the Cladis R package to date the metatree. We've recently used the metatree to investigate the influence of ancestral body size on ecomorphological trends in synapsid radiations, and I'm going to briefly talk about that as well, because we're just about ready to submit that work. Synapsid history extends 320 million years and is marked by iterative evolution of ecomorphologically diverse clades, culminating in extant groups, the monotrines, marsupials, and placentals that display spectacular morphological and ecological diversity. The conventional wisdom states that throughout synapsid history, small-bodied faunivores are dominant early on in evolutionary radiations, and they give rise to ecologically diverse clades. Previous work has shown that small-bodied faunivore is the dominant ancestral ecomorphotype early in mammalian radiations, mammaliform radiations, with the pattern of increasing size in crowned mammal groups often used to test Cope's rule. However, there has been no rigorous test of whether this pattern applies across the entire evolutionary pattern of synapsids. So we tested whether small-bodied faunivore is the ancestral state of lineages that gave rise to major radiations within synapsida by examining macroevolutionary patterns in body size and diet during major synapsid radiations. In the past, this hasn't been possible to test this hypothesis because we're lacking the necessary infrastructure, namely a comprehensive synapsid phylogeny. But now we have a handy dandy metatree of synapsid species from the Carboniferous through the Eocene that we can use. So we use that metatree in combination with jaw lengths that we used as a proxy for body size. And then dietary reconstructions for 404 synapsid species to investigate body size and dietary evolution during these major radiations of synapsid subclades. Dietary information we got from the PBDB and literature, and then the diets were then collapsed into 3 groups: the omnivores, faunivores, and herbivores. For our primary analyses, we analyzed 5 major synapsid radiations. So the pelycosaurs, therapsids, cynodonts, mammaliforms, and therians listed here. And each of these radiations is marked by considerable ecomorphological diversification, with descendant lineages achieving a broad diversity of body mass and diets. My computer needs to think about this for a minute. There we go. To quantify trends in body size and diet across clades, given phylogenetic position within each clade, we regressed body size and diet against patristic distance. And patristic distance provides a measure of the phylogenetic proximity of each taxon to a selected node within a phylogeny. So we calculated patristic distance as the sum of the branch lengths in units of time from each taxon to our sample, or in our sample to our outgroup taxon. Synapsid body sizes regressed against patristic distances indicate that there was a clear bottleneck in body size among mammaliforms, with greater variation in size among taxa with shorter and longer patristic distances compared to the mammaliforms. To understand the degree and direction of body size evolution in more detail, we also calculated the ancestral state of each major ecological radiation. Reconstructed ancestral jaw length, shown in the bar graph here, was shorter for each successively more crownward ecological radiation, with the exception of pelycosaurs having a shorter ancestral jaw length than therapsids and cynodonts. From ancestral lineages of non-mammaliform synapsid radiations, descendant taxa evolved towards both larger and smaller body sizes, although they generally do not approach the extreme small size observed in some mammaliforms and mammals. Let's think about it again. There we go. So regressions of quantitative diet assignments, regressions of quantitative diet assignments against patristic distances indicate a mammaliform funivore bottleneck that's present at the same phylogenetic positions as the body size bottleneck, again with greater variation among taxa with lower and higher patristic distances compared to mammaliforms. There we go. There was a trend from faunivory only to faunivory and herbivory in each of the major ecological radiations and the lowest regression slope among pelycosaurs. We also fit several evolutionary models to our data, and the best fitting model was a 4-peak Ornstein-Uhlenbeck model with trait optima that vary among pelycosaurs, therapsids, cynodonts, mammaliforms, and therians. Here we go. It's taking a minute for my computer to think about this one too. The best fitting models also describe the shift in body size evolution from pelycosaurs and therapsids to cynodonts as well. So with a few exceptions, the range of body sizes within cynodonts is more constrained to the medium and small sizes of pelycosaurs and therapsids. So this may suggest selection against larger cynodont body sizes or an inability of cynodonts to invade ecological niches that require or accommodate larger body sizes due to the ecological dominance of reptiles at the time. There we go. So the shift to small ancestral body size is one of several aspects of the mammalian phenotype to emerge in the late Triassic. And may stem from a reduced extinction risk of small-bodied faunivores. Additional aspects of the mammalian phenotype are listed here. The alignment of these smaller trends toward phenotypic attributes listed here, other than body size, as well as early trends towards nocturnality and fossoriality, most likely lies behind the strong small to large body size trends seen in the mammaliform and therian ecological radiations. Many mammaliforms and therians were already at or near their absolute minimum body size during the Jurassic and Cretaceous, and the lower bound of that is determined by physiological constraints. The absolute minimum body size for mammals acted as a reflecting boundary, and thus larger body sizes represented the only available morphological space into which mammals could evolve. Many studies have argued that mammals were restricted to small body sizes during the Mesozoic because of competition with or predation by the dominant dinosaur fauna. Once niche space opened up and or predatory constraints were lifted after the KPG extinction, mammalian body size diversification consisted of passive trends toward larger sizes. In this way, the end Triassic body size bottleneck permanently altered macroevolutionary dynamics for the remainder of synapsid history, although this change did not fully manifest itself until after the KPG extinction, when about 140 million years later. So thinking, here we go. The law of the unspecialized states that unspecialized species have a reduced risk of extinction compared to highly specialized species, suggesting that there isn't necessarily an active trend toward larger body sizes or more diverse diets, but simply a passive trend away from less specialized taxa. In synapsids, if relatively small faunivores or omnivores are less specialized than larger taxa, especially those with herbivorous diets, then they may be more likely to survive challenging ecological or environmental stresses like mass extinction events, subsequently diversifying and refilling ecological niches. Our data doesn't fully support the law of the unspecialized because early groups aren't small, nor do they start out small. The conventional wisdom is that progenitors of synapsid radiations are small-bodied faunivores, and that made sense in the view of the law of the unspecialized. But now that we bring in early synapsids to the picture, we see that the conventional wisdom doesn't hold up. Those radiations are nested where subsequent radiations have their own key innovations enabling the radiation. The progenitors of each radiation may seem unspecialized, but only in hindsight. So as an example, small-bodied flaunivorous mammaliforms, that's a mouthful, fit the bill for stereotypical ancestral forms of later mammalian species. But those same small-bodied flaunivorous mammaliforms look pretty specialized when you consider earlier cynodonts and therapsids. Okay, so that was the first sort of job I have on this NSF grant, was making this big meta tree. The second job will be doing geometric morphometrics, or turning shape into math, for all of these four-limb elements in synapsids. And we're doing this in 3D with 3D surface scans of every non-mammalian synapsid we can get our hands on in museums all across the globe. And thankfully, we've mostly finished that up right before the pandemic struck. This is just a small claim to fame that I have for this project, is that right before the pandemic, I went to a conference in Australia and then flew back home. The next day I went to South Africa to start scanning these guys. So when I first started the museum, I ended up in 5 continents over 5 days, which I don't really recommend, but it was pretty fun. This is just a short video showing how this surface scanner works, mostly because we're going to have one at the museum soon, which I will tell you about in a moment. And so this is Jackie Longmiss, a recent PhD graduate from Chicago, and she's been here at the museum for a long time, until recently, using this white light scanner to get a 3D mesh in real time of the whatever object you're scanning, so in this case, the skeleton, and then it also takes a color image and wraps it around that mesh. So you have a lot of information in the 3D result that you have digitally. Okay, so now we get fancy. So instead of just putting landmarks on a 2D object or just expanding that to a 3D surface, every time you put a landmark down, you're describing the shape in that actual spot. And all of the space between landmarks is missed. And so you can't record shape and you can't figure out how that shape is changing from animal to animal. And so we have to get fancy. And recently there's been some new work on using surface sliding semi-landmarks with projection from a template, which is, it sounds really fancy, but I'll explain how it works. Basically, there are 3 different kinds of landmarks. So there's the regular homologous landmarks that you can put on points you can find on every single animal, and those are in red here. And then there are semi-landmark curves in these yellow landmarks that act as a fence that fence off homologous regions of the object or bone, in this case, that you're interested in across all of the different animals. And so those 2 kinds of landmarks, the homologous landmarks and the semi-landmarks with yellow fences, are placed manually over all of the bones that you need. And then you also place them on this template. So on the far left here, you can see for this version in this awesome paper that came out several years ago now, talking about generally how to do this technique. So you place those homologous landmarks and the fences on the template shape. And then you also place a whole bunch of surface semi-landmarks in blue that are confined to each one of those fenced off spaces. And then you take your template and you pretend that it's made out of kind of stretchy balloon material, and then you wrap it over each one of the bones that you want. And then wherever those blue landmarks fall, when it wraps itself around the bone, that is the coordinates that you take. And so you get a whole list of coordinates, just like I was talking about before. But now you get a better sense of how the overall shape is changing from animal to animal. So that's our plan coming up. We have all of these scans taken. We're making sure that they're good to go for landmarking, creating those templates, and now we start landmarking them. So now that we have sort of the time calibrated phylogeny, we have some 2D shape data that Jackie Longmiss has been working on for her PhD. We have 3D shape data that we're finishing up now. And then we will also be finishing up muscle and joint reconstructions with our team over at Harvard. We can answer a lot of really interesting evolutionary questions about why mammals can do all of the amazing things that we can do with our forelimbs. Okay, so the last thing I'm going to talk about is some work we're going to be doing in the near future on synapsid jaw and middle ear evolution. The homology of mammalian middle ear ossicles with the postdentary bones of their ancient ancestors represents one of the most remarkable evolutionary transformations in vertebrate history. Over roughly 150 million years, these bones changed their shape, location, and function, resulting in an iconic mammalian morphological feature seen in no other group. And it's a prime example of gradual evolution of complex systems. The definitive mammalian middle ear is unique in its functionality. It is highly sensitive to broader range of sound than other groups. It also has, so it's highly sensitive to a broader range of sound frequencies than the tympanic ears of other tetrapod groups. And that's due to better impedance matching and more evolutionary flexibility made possible in large part to the incorporation of these postdentary bones into the middle ear. The fossil record and embryological data have shown how mammalian middle ear evolved, but why the synapsid lineage was the only tetrapod clade to incorporate parts of the mandible into the middle ear remains virtually unstudied. So we're doing a first of its kind study in the near future and using this cutting edge 3D imagery technology that I showed you with the white light scanner, along with these sort of fancier new geometric morphometric methods to investigate if an early adaptation to mandibular bone conduction of sound, so putting your jaw on the ground and sort of listening to vibrations that way, in these basal synapsids made a really tight connection or integration of traits between the postdentary bones and the mandible. Whoops. That continued through time. So it took a really long time in order for those postdentary bones to break off from the mandible in mammals. We recently received funding from the Granger Bioinformatics Center here at the museum to purchase the white light scanner. It's a surface scanner to do all of this work, and I can't wait to get started. We used to need 2 different scanners to do this work because of the size range of the things that we were scanning. And then those scanners that we used for our NSF grant are actually owned by the people at Harvard, so we don't have ready access to them. What's really exciting is that these new white light scanners have changed slightly so that we can get the whole entire size range that we need of these bones in just one scanner. And then now we can have it at the museum for us to use instead of having to continually borrow one from Harvard. So we're excited to get going on that project. And very lastly, thank you to all these people that have helped with all of this work, from my PhD work up through my postdoc work. We've had funding from various NSF grants, along with some funding bodies at the University of Indiana, where I did my PhD. Thank you very much.
All right, thank you, Spencer. So please enter your questions in the Q&A area, and we will be sure to try and go through those. First, I see there is one question in the chat. We can start with that while people are adding their questions to the Q&A. So Kelsey Stilson asks, can you speculate on how mandibular bone conduction of sound and other information correlate with reduced dental series, so diphyodonty or monophyodonty, or if there's even a connection?
I don't actually know a whole lot of details on that. Ken, you might actually be able to answer that better than I can.
Yeah, I guess I would say that some of the parts of that transition happen at a similar time. So kind of the emergence of something that is close to the definitive mammalian middle ear is more or less in place by around the Jurassic, also around the time when diphyodont tooth replacement pattern is happening. But I guess the transition begins, or like repurposing of the post-entry bones begins well before that in animals that still had kind of a more like kind of stereotypically reptilian continuous tooth replacement process. So I don't know if they're necessarily directly related to each other. Right. So while we're waiting for other questions, Spencer, I actually have one for you about the bird work. So it looked like one of the sort of consistent modules you were recovering across the different elements was at the joint surfaces, which kind of makes sense functionally, but I was wondering if you have any information about whether there's any developmental reason to expect that in terms of how the sort of bones are patterned themselves.
Not necessarily like clear developmental evidence. It's more of a functional reason, I think, like you were saying, but not only for flying. So that's the obvious functional reason for why the joints would be important. But once you go flightless, that's kind of still there. And so you're not flying anymore. You would think that that would kind of break down pretty quickly, but it still has a functional reason. So when imagine you're a bird and you gotta walk around, you have to pull up your wing pretty tightly, because if all of that joint and the proportions between the individual limbs get messed up too much, you won't be able to fold up your wing, and so it's just going to be dragging on the ground, and then that hinders your movement, plus infection and all that jazz. And then even at the beginning of the talk when I showed the chicken egg, it's kind of the same thing. Like you have to be able to fold up and squeeze in there, and so even though you're not using your joints for flying anymore once you list, you still have to fold up your arm as long as you keep your arms.
Okay, thank you. So I guess we'll wait another moment to see if any questions arrive. Otherwise, you may well be off the hook.
It's a good teach a dinosaur evolution class at 1.
Yeah, I guess while we're waiting, just in terms of general information, so Spencer mentioned that we're going to hopefully be purchasing the surface scanner soon. We have a meeting with a representative about that coming up next week to kind of begin that process. And once we sort of get it in hand, we'll be setting up a means for people who are interested in using it to be able to sign up to do so, because we would like to make sure that it is a useful resource kind of across the museum and it gets used as much as possible. All right, so a question has appeared in the Q&A. This is from Bill Simpson. How does the adoption of upright posture correlate with reduced acquisition of sound waves via the head touching the ground?
Yes, so you would think once you sort of push up off the ground, your jaw isn't that close to the ground anymore, and you don't necessarily need to be listening through the ground. But if you're fossorial and you're digging around in there, especially if you need to hide from predators, you can still very easily sort of crouch down and place your jaw in there to figure out what's coming next. And so you still could be using that for longer than you're lower down on the ground.
Yeah, and I guess one thing to note is that there's actually some mammals today that still have an aspect of so-called jaw listening that they do. Okay, so a question from John D'Angelo. What are your thoughts on whether the tympanic middle ear is homologous between mammals and reptiles?
Basically that it's not, and it's analogous. It's evolved a couple of different times as far as I'm aware, and the structure, sort of how that works and the elements involved and the placement of the tympanic ear is all pretty different every single time that it evolves. And there's quite a bit of talk and controversy and like exactly how that works up to having a tympanic ear in mammals as a clade. But between all of the groups, it's just sort of analogous evolution of the same functional tympanic ear.
All right, and another question from Ellie Zach. Is there any correlation between ecology and use of jaw listening?
It's kind of hard to speculate on that with the fossil mammals, other than to say that if you're fossorial and you're digging around, it's useful to have that jaw listening. As Ken mentioned, there are some mammals today that use jaw listening, and those guys are close to the ground and need to be digging around and hiding, and so they use their jaw for listening too. So we can sort of speculate that they had sort of that similar function back in time.
All right, looks like a new question from Robin Watley. How do you correct for distortion in fossils with the morphometric landmarks if the fossils are particularly distorted from preservation, for example?
So mostly you don't, because you can technically do that to some degree, correct for distortion, but it is really time consuming and difficult to do that, and the whole process is already time consuming. And so mostly people don't do that. So when I'm looking through all the fossils, if something is broken, or especially if it's really broken or very distorted, or if it's like a young animal, etc., I mostly don't use it. Thankfully, we have enough specimens that it's not too much of a worry when you have to throw out those occasional specimens. When it gets really bad, so for an example, part of my PhD work that I didn't talk about is with non-avian theropod dinosaurs and their limb bones. And when you need multiple limb bones from the same limb, from the same side of the body, from the same individual across a bunch of different species, that far back in time with fossils, you end up with kind of a crappy specimen list. And so statistically, it's just not great to be able to just leave out all of the crappy specimens. So instead, you sort of look at what you have, and then you make sure that your landmark configurations don't take into account those areas of the bones that are usually messed up or broken off, if at all possible. Sometimes landmarks are not the best way, or like the best thing to use to get that same kind of information. So if your set is really bad, instead of putting a bunch of landmarks on there or landmarks at all, you can just go back to regular old linear measurements. It just depends on the data set that you have. So, but yeah, unfortunately, you can't really make sure that they become undistorted digitally and then put the landmarks on.
Yeah, doing the retro deformation processes does weird things to covariance patterns in the landmarks. It's something that we encountered with the work we've been doing on synapsid vertebral columns. We actually got a retro deformed diadected amnio vertebral column CT data set from some colleagues, and tried to include it in our analyses of regionalization, and got really, really bizarre results that I'm convinced are because of basically not being able to cleanly remove a deformation signal from kind of the biological variation that's actually there. So like Spencer was saying, it's very problematic. So there's a question from Lo that's come in. Can you think of some parallel examples outside of synapsids where diversifications start with ancestors of small body sizes?
Well, it's the first one to come to mind, I guess, is dinosaurs that start out sort of as little guys and then get larger and larger. And again, it's not necessarily, even in those kind of groups, always an active trend towards a larger body size, but just it's a space that's not explored at the beginning of the variation. And so that as evolution happens, things get bigger and some things stay small. And so you get a wider sort of diverse range of body sizes. So it happens in lots of things, not just mammals, but some more modern mammals are the classic example of that type of variation happening. I saw a question in the chat actually about what mammals do the jaw listening, and that fact has fallen out of my head at the moment. Ken, do you remember what mammal does that?
I actually would have to go back and look at the grant proposal where we mentioned it. I think that it is some fossorial species, and it may be fossorial rodents, but I'm not 100% sure off the top of my head. So yeah, sorry about that.
Yeah, I'm just curious.
Yeah, so another question from Lo, where are examples among non-mammalian synapsids, or sorry, non-synapsids, where diversification happens with large-bodied ancestors?
I have to go through my animal knowledge in my head now and pick that out. Ken, can you think of any off the top of your head?
I guess part of the issue is what counts as like large size. Because
true.
like therapsids start out at I guess what I would call sort of a medium size compared to like particularly mammals today. So animals that are sort of like probably dog to wolf size. Although that's obviously not huge compared to like the extremes that like dinosaurs or even modern mammals reach. But yeah, I'm thinking of like a really large ancestral form. Nothing really leaps to mind.
Yeah, I mean, and especially sort of the obvious and like direct trend downward to small sizes is hard to pick out. There are definitely some things like in some squamates that end up, you start out with a larger ancestor and then you can, some of them get down to a smaller size, but a trend overall down to the larger size is hard to pick out in other animal groups. It's pretty rare. And we weren't necessarily looking to find it in synapsids. It was kind of cool that it showed up early on, that they don't all have this trend from small to large.
Yeah, I would say the sort of early synapsid trend is sort of starting out at sort of like mid-size and then expanding both towards smaller and larger sizes, which kind of makes sense as like a diffusion process. And then, like Spencer was saying, you have the sort of end Triassic size bottleneck that kind of changes things and sets you ancestrally at that smaller size that then causes sort of the trend for increasing size and subsequent radiations. All right. Any other last minute questions? All right. If not, Spencer, virtual round of applause. Thank you very much for doing the talk today. And yep, good to see
Thank
everyone in some sense. And have a good day.
Thanks, you too.